Ladies and gentlemen, it's my great pleasure to chair the third BISMiS virtual meeting. Today, the president of our BISMiS, Professor Dr. Kamlesh, he's busy for another meeting, but anyway, he will join with us and attend our discussion later. And I will chair this talk. Today is my great pleasure. Our BISMiS to the third talk. We will invite the Professor Juncai Ma from Institute of Microbiology, Chinese of Academic Science. Before the formal talk, I will briefly introduce the Professor Ma and also the brief introduction of BISMiS. Okay, the first I will take a few minutes to introduce the speaker, today's speaker, Professor Dr. Juncai Ma. He's received his PhD degree from Department of Bioresources of Meiji University in Japan. Now he is the director of Microbioresources and the Big Data Center in Institute of Microbiology, Chinese Academy of Science. He is also the director of China National Microbiology Data Center and also WFCC World Data Center for Microorganisms. The abbreviation is Wdcm. And he is also a board member of World Federation of Cultural Collection, WFCC. Today, his topic is he would like to introduce the GCM 2.0 WDC 10,000 sequencing project for our type strains. Now it's time to invite the today's speaker, Juncai Ma, from the Institute of Microbiology, Chinese Academy of Science, will give the today's talk. Okay, Professor Ma, please.
Okay, thank you very much for Professor Lee's nice invitation, and also thank you very much for your nice introduction. So, can you see my PPT file?
Yes.
Okay.
Yes, it's good.
Okay. Today is my great honor to have this chance to introduce our WDCM GCM global catalog for microorganisms, taken 10K sequencing project for sequencing for the type strains. So the target of this project is closing the genomic gaps for the validly published prokaryote species. I'm Juncai Ma, coming from the World Data Center for Microorganisms and also China National Data Center for Microbiology. So now there are about more than 800 microbial culture collections from the 78 countries made their registration in the WDCM. So we have close communication with the culture collections worldwide. So from 2012, we started international cooperation. We call it Global Catalog for Microorganisms. So we call each microbial culture collection worldwide. You're welcome to provide your catalog information to us. Then WDCM developed the global catalog for the public. So now we have more than 133 collections from the 50 countries join this global catalog cooperation, and about 480,000 strains information in our global catalog. So in the right side, you can see, so for any of the user, if you input a particular scientific name, such as Bacillus subtilis, then you can get information from worldwide. Which culture collection can provide the Bacillus subtilis strains to you? Then you can get the you can choose the isolation information about the strain was isolated from soil, from water. And if you want to buy the Bacillus subtilis strains, you can select you want to buy from the Asian collection or European collection. Then you can get the strain information about isolation, taxonomy, and also the paper, global paper, patent sequences information related with this particular strain number. And from the 2017, we start a new international cooperation. We call it GCM, Global Catalog for Microorganisms 2.0, next generation of the global catalog. So we hope in the future, global catalog can have not only the catalog information, but also genome information and more information. So we started this 2.0 cooperation sequencing for the texture. And our target for this sequencing project, we want to do the sequencing of 10K texturants for the bacteria, for the fungi, and so on. So Wdcm will cover the cost of the sequencing, database development, and the data analysis. And all the data will be opened to the original culture collection and also to the public. So we welcome to any of the microbial culture collection worldwide. If you have texture, you're welcome to send the DNA or strands to us. Then we do the sequencing for the texture, and then data will be opened to you and also to the public. So this is a project we designed. So through this project, we want to provide the more and more data, more and more information for the taxonomy science, and also for the gene discovery and notation, and so on. So, and also we want to provide phylogenetic information of the metagenomics data, and also we hope so through our database, the scientists can make a better understanding for the relational mechanisms and the relationship between the phenotype and the genotype for the microorganisms. So we want to provide more and more data for the type strands from our Wdcm. So now we can understand through this PPT file, we can understand in 2015, in the world, there are about 12,000 type strands. Among them, about 400 of them are sequenced. And in the 2018, about 14,000 type strands, about among them, 6,000 of them are sequenced. So by the now, we can understand for the JGI, they start the nice GBA project since 2007. And in the 2017, they published a nice paper for about 1,003 reference genome. And from the right side, from this table, we can understand from each sequencing center, how many data or how many sequences they have finished for the test strand. So from JGI, they published about 3,000 type strand genome, and from our Wdcm, now we opened about 1,500, and also you can see the information from the other sequencing center. And we can, from this PPT file, we can understand that from about the 2014, every year about 800 new species are published. Among them, about 1,600 to 1,800 type strands are there. And from this PPT, you can understand who are the top 10 collections who host the TAP strands. For the red color, you can see the Asian collection now play more and more important roles to host, to deposit the new TAP strands. And from this PPT, we can understand, we have the list from each microbial culture collection, how many TAP strands they have. DSSA is the number 1, then JCM, ATCC, TCCM, MBRC, and so on. And we understand the top, there are about 66,000 TAP strands deposited in 134 microbial culture collections worldwide. Among them, top 30 microbial culture collection deposit in the top 30 microbial culture collection, they have more than 90% of the TAP strands. And in our project, so we have the list how many microbial culture collections they join our GCM sequencing project. So now about 26 collections from the 18 countries join this sequencing project. Some of them provide DNA, some of them provide the strand to us. Then we do the sequencing works in Wdcm. And we also arrange the international working groups for the bacteria selection, fungi selection, SOP, database development, and also IP, and so on. So it's a very international cooperation project, international contribution, and the strain and the DNA from the microbial culture collections worldwide, and the working group members are also international. And for the strain selection, we focus on the phylogenetic diversity, scientific target, and availability of the strands and the non-importable safety. And also, recently, we are very interested about the, for example, 2 TAP strands, same TAP strands in the different culture collections. So we also want to do the sequencing for the quality or something, right? And through our project, we developed several standard documents, MOU, MTA for any of the collection. If you want to join the project, we need to sign the MOU with your host organization. We also need to sign the MTA document with you. And through the international working group, we also developed several SOPs for this project. And also, we developed the TAP strand sequencing protocol and the data analysis pipelines from the strain reculture, DNA extracting, library, assembling, and so on. So everything is standardization. And here's the list, how many, for each collection, how many DNA or strands they send to us, and how many of them are finished for the sequencing, how many of the strands is on the way. So we have finished about 1,500 strands for the sequencing, and another 2,500 now is on the way, it's on the development. And we also developed the high quality reference database, 16 RNA quality control method, 16 RNA gene and whole genome database, and our data is opened to the public. And from this PPT file, you can get the comprehensive information of the all valid published species. So you can, from this table, from this picture, you can understand in the different taxonomy level how many species we have finished, we have sequenced. For the red part is the species that from Wdcm we sequenced in the GCM 10K project. And here is our data portal. So the address is gctab.wdcm.org. So through this website, you can understand through our WDCM database about 16,000 genome data worldwide available in our database. About 66 number of the type strands, and about 12,000 genome. And from our WDCM side, we finish about 5,049 type strand sequencing. And through our website, we provide the data services. So you can search of the species, you can browse the type strand from each culture collection, how many strands they provide to us. And also you can get the table in the right side. For each species, you can understand for this particular species, how many type strands are there, and in which culture collection the type strands located, and for each particular type strand, which sequencing center finished the sequencing work for this particular strand. So for some of the strands, they are not sequenced. So we can show you this kind of the nice information. And also we provide the data analyze service to the original microbial culture collection who provide the DNA of strand to us, and also the data analyze is also opened to the public. And you can also, through our website, you can understand the information, strain information for the type strands that we sequenced. You can get strain information, species information, and the taxonomy information about the particular type strand. And for the strain information, we also list the phenotype information for the, for example, for the oxygen, we have 9 types. For the sheep, we have the 11 types. So we also list this kind of the phenotype information. So in the future, if you want to study for the relationship phenotype and the genotype, you can work with us. You can or you can download data, so you can do your own research. And for the sequencing result, so you can get this kind of the information about the GC, about N15. You can get all this kind of the sequencing result for each particular sequencing data. And also you can get this kind of the information and image file and so on. And also we provide the online genome assembling annotation, and also new species identification. So you can input your own sequence data for a particular new species or strain, then you can get the identification result using WDCM database system. And also, this is an international cooperation. We are not only do the work in our own data center, but also we are making the cooperation with the other international organization. For example, we have signed official MOU with Wdcm, with Budget Money Trust. So we hope in the future we can have WDCM can do this kind of function. So we welcome the send. You can send your research proposal to us. For example, you want to do a particular research project for example, a study on a particular environmental or particular function of the microorganisms. Then we can send your proposals to the cultural collections worldwide to invite cultural collections join this particular joint research. So if the collection is happy to join, we will ask the collection provide the strengths related with this particular joint international joint research. And the WDCM will do the sequencing for the strengths, data sharing, database development, and we work together to do the data analysis. So we will try to use our global network for the cultural collection to do not only the sequencing project, but also for the future joint study work. And for the official MOU with IJSEM, so we agree for any of the scientists worldwide, if you want to publish a paper for the new species in IJSEM, you are welcome to send your DNA or strength to us. WDCM will do the sequencing free of charge. We will not charge the cost of the sequencing, and the data, the sequencing result will return to you, the science, and also the data can be showed to the IJSEM editor. So editor can see the data automatically through the WDCM database for your strength when you submit your paper to IJSEM. And also your data can be automatically transferred to ISDC. So your data can be available in DDBJ, GenBank, EMBO, and NCBI, and so on. So we write a paper to introduce these WDCM particular services, and this paper is published in IJSEM. And also now more than 170 species are sequenced for the science they contacted us. And also we have the cooperation with the Belgium Money Trust. So for example, for this particular family, through our WDCM database, we can understand for this particular family, there are about 78 species. And among them, 29 of the species are sequenced. And we can clearly understand among them, for the non-sequenced type strands, in which cultural collection they host, they keep, they deposit the type strand. So we can contact our cooperation partner of the cultural collections to invite them join this joint research. If you are happy, you can provide the type strand or DNA to WDCM. WDCM will do the sequencing for this joint research. And in the future, we welcome for any of the scientists that you can provide your new proposal joint research. We have a scientific committee and working group, so we will discuss about your proposal. Once your proposal is accepted, your proposal will be sent to the cultural collections of our cooperation partner. And if the collection want to join, they will provide strands or DNA. Then WDCM will do the sequencing, we will do the assembling, we will do the data, we will work together to do the data analysis, then we will work together to make the joint publication. And also all the data is opened to the cooperation partner, and also when the paper is published, our data will be opened to the public. So we welcome this kind of cooperation in the future. So finally, I would like to thank the great contributions from the cultural collections worldwide. So they provide DNA, they provide the strength. So it's a great contributions from the cultural collections worldwide. And also I would like to thank the great financial support from the Chinese academic sciences, and also from the Ministry of Science and Technology from China. They give the great support for Wdcm activities, especially great support for our sequencing project. And also, I would like to thank the great work, the hard work of my team. And in the future, if you have you want to contact to us, so you can contact my colleague, Dr. Lin Huan Wu. So here is the email address of Dr. Lin Huan Wu. Okay, thank you very much.
Guys, can you continue to chair the last session?
Thank you, Professor Juncai Ma, for such a wonderful talk and sharing the details about the GCM and how people can contribute to this grand proposal and the project that you have undertaken. There are a few questions that have come up, and I would like to begin by asking, one very peculiar question that has been bothering Indian researchers is the NBA rules. The Indian researchers are not able to submit their cultures outside of India. And this question has been asked by, Ratan. And the question is basically any plan to take biodiversity board into this umbrella. Otherwise, it is difficult to send out strains for deposit to culture collection centers outside the country. So Indian biodiversity laws do not allow sending Indian origin cultures outside of India. So how are we tackling this? And it is not only India specific, there are many other countries that are facing the same issues. So even if people want to share their cultures for sequencing project at WDCM, how are we tackling this issue?
For the WDCM, when we receive, when any of the collection, when you want to join this 10K project, we will sign the MOU, we will sign the MTA. So we will ask you need follow your national laws, especially national law related with the CBD and also NAIA protocol. So I'm also the board member of the WFCC. So we understand the situation of the India, Brussels, and so on. So you have your national law for CBD and the NIL protocol. I don't know, in case you cannot send the strain to the other country, whether you can send the DNA to the other country.
Okay. So instead of the strain, the DNA can be sent.
Yes. If you can send the DNA of type strands to us, we can also do the sequencing for your type strand.
Okay. All right. Amit would like to add an answer to this question because we've been working quite hard for, getting these permissions from the NBA. So Amit can add at least from the Indian perspective. Amit?
Yes, to answer Ratan questions and to add answers to Professor Juncai Ma, the first thing is that we can submit cultures to culture collections outside India. There is no restriction on that. We can also deposit DNA for research purpose outside India. There is no restriction on that. The only thing that one has to follow a procedure by applying it to National Biodiversity Board. National Biodiversity Authority. So there is a prescribed format, there is a prescribed form where you can apply with the strain. ID and all other details that you need to fill in. And you need to also state the purpose for which you need to, the reason why you need to submit that culture for a particular purpose. The only caveat in this entire procedure is that the foreign culture collections or foreign entity for that matter cannot circulate that particular strain without prior approval from the NBA of India. So any Indian researcher, a registered one associated with any official organization, can definitely send the cultures abroad with prior permission and prior procedure to be followed. That's the addition that I wanted to make.
Okay, thank you, Amit. So, Professor Ma, there is another question that has come up, is that if a person submits a proposal to get some sequences, so one question has come from Humera, and the person is asking, are we charged for the sequencing that is carried out at WDCM?
How to say, if you are the cultural collection, if you have type strengths, if you join the WDCM 10K sequencing project, when you provide the DNA or strengths to us, we will do the sequencing. It's totally free of charge. So second case is if your purpose is want to publish the paper in IGSEM, because we have the official cooperation with IGSEM. In this purpose, when you send your strand, your DNA to us, we will do the sequencing for you. It's also free of charge.
Okay. So in both scenarios, there is no cost, even if you submit as part of the 10,000 project?
Yeah,
or
because our WDCM project, basically now we focus on the type strand. If you want to do some joint work related with the type strand, we can do the sequencing for you. It's free of charge.
Okay. Now, I would like to ask my own question. I'll get in there, take the liberty. How does the sharing of information work at WDCM? Because I presume DSMZ is also running their own sequencing project where they are doing the type sequencing, the sequencing of type strains. And for them, the database is available online and they can share, anybody can download the information that is available. Does it work the same way?
Yes, yes. For the WDCM, basically we have two steps to share the data. So first, when we receive DNA or strands from the original cultural collection, we will do the sequencing. Basically, we will finish the sequencing in 2 or 3 months. Then we will immediately return the data to the original cultural collection. So original cultural collection can open the data in their own homepage or catalog. And for the WDCM, we will make the joint paper with the original cultural collection. So when we publish paper, all data will be opened to the public. So now we have opened, I think, more than 1,000 sequence data.
Okay. There's another question that has come and it is very specific. Is there a protocol for sending samples from a country? This has been asked by Anila.
For this particular question, I think you can contact my colleague, Dr. Lin Huan Wu. So she will send detail information for that.
Okay. Another question is, can Ectinobacterial strains be submitted for sequencing? Obviously, this question pertains to somebody who is working on Ectinobacteria. Sunita Bunde.
Yes. For basically all kind of the type strands, we will work on.
Okay. I presume Wen-Jun is submitting a lot of cultures since he works on Actinobacteria
Yeah.
quite a lot.
Yes.
Am I right, Wen?
Yeah, you are right.
Okay. And one last question from my end is going to be, how does the access to the database work, Professor Juncai? Because if Wdcm is conducting the sequencing, can we also download the raw data and do the annotations at our end, or is it only the refined data that is available?
Sure. I think you can go to the Wdcm homepage to search the data and also to download the data. If you have any questions for the future questions for the data policy and so on, so you can contact my colleague, Dr. Wu.
Okay. Another question that has come up, it's kind of, more scientific. What is the basis of considering a given culture is a novel species, and how do we define and submit it as a type strain? This has come from Prakash.
Basically, if you're for our sequencing project, we need to get the officially validate, publish new species. So basically, for the bacteria, if your paper related with the new species published in Wdcm, you will be asked to deposit your strands in the two public available culture collections. So in this case, we will contact the collections for the sequencing project, and for the scientists, if you want to publish the future new species before you deposit your strands to the two collection, so you can contact us. So we also have the list for our cooperational culture collections. So you can not only send the DNA to us, but also you can easily deposit your strands in our two cooperational culture collection. You can quickly get the strand number for your paper purpose.
Okay. Anila has one more question. Can we send Borrelia DNA as it is an emerging zoonotic disease? I think Professor Juncai Ma has already answered this.
For DNA, I think it should be no problem to receive.
One question, it's more of a technical question, and it's more about the database. It's come from Dibyendu Khan, and he's asking how this data is arranged sequentially in Burgess manual. So it's more of a, yeah. How is this genomic data arranged sequentially in Burgess manual? And I think it's part of a classification question.
We can have the discussion for the data cooperation with the virtual manual.
Okay. All right. I think there are no more questions. When anybody amongst the panelists would like to ask any question is more than welcome.
Okay, Kamlesh, I have one question to ask Professor Juncai Ma. Okay, your project proposal and project has passed almost 2 years. Are you satisfied with the progress for your sequencing results?
For the sequencing project, I think now more than four years.
Yeah.
Yes, please.
In your opinion, how is the feeling for the progress? Means the speed for the sequencing for your type strain project. It's good or slow, too slow or too fast than you are expecting?
I think it's how to say, in the first 1 year or 2 year is slow.
Yeah.
Because in the first 1 and 2 years we only receive the DNA. So the original, for the first 2 years, the original culture collection can only first do the DNA work by their own staff. Then they send the DNA to us. But for many of the culture collection, they have the limitation of the staff numbers.
Yeah.
They have so many staff to do the DNA related work. So we receive the quite small number of the DNA. But now we can receive not only DNA, but also cultures. So we can do the culture DNA works in the Wdcm. So now we are very happy to have the better speed for the sequencing project.
Okay, thanks. Okay. I got.
So Professor Ma, one more question has come from Alessandro Costa, and he wants to ask, how do you manage cyanobacterial genomes that are problematic to purify?
I think I need to ask my colleague.
Okay. Okay.
So
You can go ahead.
So you want to ask how is our quality control process for all the types in genome, right?
No, I think the question is very particular related to cyanobacteria, because cyanobacteria is typically mixed cultures, and the person wants to know how do you purify or how do you manage the quality control, especially for cyanobacterial genomes.
Yeah, it is a question. It is question in our quality control process. We have a whole protocol to after we perform assembly, and we will check whether it is there several contamination contains in our assembly based on camera search and a lot of program. So after we run all of this procedure, quality control procedure, and the contamination will be removed.
Okay.
Yeah.
Okay. Professor Junkai or Shi, can you share some more details of how to submit the proposal?
You can contact Dr. Lin Huan Wu.
Okay. Okay. All right. So I think all the questions have been answered. Thank you so much, Professor Ma, for this excellent information regarding getting your type strain sequence or submitting and publishing new strains, novel strains. Thank you so much for this, very informative session. And thank you, Dr. Shi Wen-Jun, for, helping us learn few more things about how GCM and WDCM works. Thank you so much. Wen, thank you for hosting this session. There are just two more things that I would like to add. From the next week onwards, the timing will be shifted by 1 hour. We will start at, instead of, let's say, for India time, if we start at 11, we will start at 12. And for China, if we are starting at 1:30, we'll start at 2:30 now. So that is a slight change that has happened based on the feedback we have received from most of the participants all over the world. And that will accommodate participants from Europe and UK to make a more comfortable time zone, and also add few more time zones into our call. The second thing is I would like to request all the participants to subscribe to the YouTube channel so that next time onwards, you don't have to register separately for each of the talk. All those participants who have registered up till now for any of the sessions that have been conducted will automatically receive information for the next talk, and they don't have to register separately for each talk. That is one of the most important additions that we have done. And lastly, if you go on our BISMiS.net website and go to the specific page on BISMiS Live, you can see the list of speakers that we have lined up for you until the end of this year. So BISMiS Live 2021 has excellent speakers that have already been lined up for you. We had Professor Phil who gave the inaugural session, then Ian Sutcliffe, and today's talk was from Juncai Ma. For everybody's information, the next talk will be given by Professor Wen-Jun Lee, who's our secretary of the society, followed by Professor Aaron Oren, then Brian Headland, then Ramon Rosello Mora, then Professor Kostas Konstantinidis, Professor Martha Trujillo, who is the editor of all the IGSEM, then Dr. Marike Palmer from University of Nevada, Las Vegas, who will be tentatively speaking on the prokaryotic species concept. So we have lined up one session every month from excellent speakers. So please use this to your advantage and subscribe to our YouTube channel for the live stream and all the sessions that have been done. So thank you so much, everybody. And thank you, Wen-Jun. Thank you, Professor Ma, and thank you, Shi, for hosting. Thanks, Amit, once again for controlling the session. Thank you, everybody.